So first of all, thanks for the organizers of giving me the opportunity to talk here, especially because it's not during the final of the Eurocup. So it's nice to see you all here. I'll talk about the work of two students, actually the master's work from Fred, who's now a PhD student in the lab, and also some PhD work from Katja, hopefully, when I get to it. And so the RNA side of my lab tries to understand post-transcriptional regulation and not splicing typically, but rather we want to go from knowing what different RNA binding proteins do to measure different aspects of post-transcriptional regulation. And there we mean mostly stability and translation. Use these type of data to actually have readouts of different stages of the RNA metabolism to train predictive models. And ultimately what we're starting to do is to build new regulatory sequences with defined properties from that. And so one assay that we are really fond of is essentially ribosome profiling, because it allows us to really understand translation and observe essentially the ribosome in action. And the typical, you'll see some variants later, but I just want to introduce you to the basic concepts here, is that you can isolate fragments that the ribosome, in this case the translating ADAS ribosome, is bound, align them back to the genome, and if your sample preparation worked well, you actually see a periodic signal there. It's because the ribosome moves 3 nucleotides at a time. And so already a few years back, we developed a strategy based on a so-called multi-taper test that actually looks at significant spectra based on this multi-taper filtering strategy that allows you to really clean up the signal and detect periodic components, even if the data is really noisy. Because of course, as any sequencing data, it's really noisy and we can't expect a perfect 3-periodic frame. And so with this in hand, we could actually do de novo open reading frame annotations from this data. So not just quantify the number of reads and known annotated regions, but rather look at what is actually translated in the genome. Where do we see ribosomes actually performing their function? And this is from the original RiboTaper paper. And you can see on the right side, just these charts where you can see essentially coding and non-coding ORFs, and especially you see that there was lots of activity in what's called upstream open reading frames. So there's activity where reading frames are, short reading frames typically are present in the 5' UTR before the main coding reading frame. We also detected lots of pseudogenes and antisense transcripts, and that's really interesting, for instance, for new antigen prediction in cancer cell lines. That's all follow-up work, you can check that out. And so this and other software is actually the foundation of an ongoing consortium that works with GenCode to annotate based on ribosome profiling data where actually used reading frames in the genome are, to complement essentially proteomic data sets, because not all of these functions of the ribosome scanning have to lead to stable protein products, right? They could lead to new antigens. These upstream open reading frames typically do not show a preserved coding information. They just show the start and stop codon being conserved. And of course, there are some microproteins and there are groups that are interested in this, but that's not really the focus of my lab here. What we rather decided to do is, or in the follow-up work a few years ago, we looked at now the presence of alternative open reading frames and transcripts. So for this, we turned to annotated transcripts, layered on the RiboSeq evidence as spliced reads. Now these are short fragments, right, 30 nucleotides, but if you have known junctions and just align them to known junctions, this is actually doable. And from there, we can quantify with an EM-like algorithm how prevalent translation on the different annotated isoforms is. And so, we detected some with alternative isoforms. Most actually just have one dominant ORF that's translated. So even though there are multiple isoforms, the translation activity typically really derives from just one dominant protein isoform. But of course, there's thousands of cases where you actually have 2 or more isoforms present. And when we looked at this in a bit more detail across different cell lines, to our surprise, we saw that 40% of the alternative open reading frames actually did not come from alternative splicing products of the main open reading frame, but actually constituted shifts in the dominant translated ORF between the upstream open reading frame and the main open reading frame. So this sort of as introduction, why are we actually interested in this? Why do we want to understand UTRs, 5' UTRs, the impact on translation, and the function especially of these upstream open reading frames? And so that they are functional, there's enough data. For instance, here, this is real data from fly that shows in different developmental stages, from embryos to larvae. You actually see exactly the switch of a dominant open reading frame being translated between the upstream open reading frame and the main open reading frame. So they seem to be really a potential source of major translation regulation. And we, in the lab, actually use zebrafish as a model system. This is a different paper, not from us. We have similar things now going where we can make reporters essentially then in fish as a nice model system to read out the effect of these individual open reading frames. Because oftentimes there's more than one and they might overlap and create all kinds of complicated scenarios, whether the ultimate eventual readout on the main open reading frame is actually positive or negative. Okay, so 5' UTRs, the median length is actually not that long. It's a few hundred nucleotides. And in some of these species, I think it just reflects the status of the annotation rather than the typical length of the UTR. But you can see they're massive long tails. And so, for instance, the initiation factors, the essential eukaryotic initiation factors, all have really long 5' UTRs with many upstream open reading frames. So it's definitely autoregulation exactly in the initiation process itself. And so turning to Fred's work, we thought, how can you start to understand this? And we were not the first ones. There's really nice work from Georg Selig. There's actually a follow-up paper to this study from 2019 that just came online, I think 2 weeks ago, where they now cloned, in this case, only 50 nucleotides. So that's the limitation of natural sequences and of synthetic sequences to in vitro transcription, polysome profiling, and then actually read out what the previous speaker mentioned was called mean ribosome load. So this is not directly ribosome profiling because you have a reporter gene, so all the reads will look the same. You rather sort essentially your different reporters, your genes with different reporters into different bins, how many ribosomes are actually on them. And that's how you get this mean ribosome load, specific now in a controlled setting just for different UTR sequences. And here I just want to make one plug. So the authors published this, and we'll see how well this actually worked. But one aspect that my lab's been really interested in and was very adamant early on to use this is to use interpretation methods for neural networks. Right? As Chris pointed out, they have millions of parameters, and we cannot understand the direct parameters, but what people over the years figured out is to look at either the first layers or the last layers to get an understanding what these networks actually do. And there's a lot of different what's called attribution methods, for instance, integrated gradients or Shapley or layer-wise relevance propagation to tell you what in your input data was actually important for the prediction task. And this might show you surprising things. So here in one of the early evaluations and image data sets, this was a task to classify modes of transportation essentially. And what these maps showed is really interesting. It's that you actually saw that it's not the ship that's recognized, it's the water that was important to classify this. Likewise here, the rails. And with the horse, it's actually not even the horse, it's the logo of the stock photo company that the horse pictures were taken from. And so we can't show these pictures enough to tell you this happens in genomics as well. It's just a nice visualization here. So what we wanted to do is to go back in these synthetic and natural human reporter sequences, retrain the model that had been published, and look and interpret, use these methods to look at what actually in the sequences was there to explain this mean ribosome load, both for the synthetic and the natural sequences. And so what you see here are these typical attribution maps. So these are 3 example 50 nucleotide sequences, and you visualize essentially with upper or lower letters if there was a positive or negative contribution to your prediction. And so if you look at the top translated synthetic sequences, so the top mean ribosome load, the main characteristic of these sequences is that they have a difference in GC versus AT content, meaning that things with lower secondary structure is more highly translated. Now, the ones that get very little translated, you can see exactly the presence of these upstream open reading frames, so of UAGs. And what we can do is from these sequences to create these meta attribution maps that show you essentially 4 ACGTs, these are the 4 rows here, what actually is going on. And you can see here even this nice, periodic signal essentially, and also a distance preference, right? Because you want a whole upstream open reading frame, so you want to start here in the beginning of the sequence so that you fit a stop codon somewhere in here as well. So far so good. Let's look at this in a bit more detail to see what's going on. So if we have no upstream open reading frame, you can actually see that there's also, if you split this non-separate classes, you can see that there is sort of the Kozak sequence showing up here, so the sequence context for initiation beyond the start codon. If you have in-frame upstream open reading frames, they're actually positive, and especially if they're close to the initiation site, because they will just provide an alternative start codon. If you have out-of-frame ones, however, this becomes negative. And if you have overlapping ones, the picture, of course, becomes a bit crazier if there's no stop codon anymore before the translation start. And so you can use these methods to gain insights really of what is actually present in these sequences. Now, these were the synthetic sequences. And if we now turn and look at how good is the model on the synthetic sequences compared to the human sequences, you can see that this is actually a really good value for this mean ribosome load. The simple convolutional neural network gets correlations of 0.93 in the synthetic data, and the model trained and tested on the human natural sequences that they also did, just cloning the natural fragments, is around 0.8. However, what you see is that if you take the AUG parts out, so if you take sequences without AUG and just retrain on the remaining sequences, the model actually really drops for the synthetic sequences, meaning that the main feature that the synthetic sequences contained were really these, by chance, upstream open reading frames. However, on the real human data, you just see a drop of 0.05, meaning these upstream open reading frames are there, but they do not contribute as much, actually, to the output as they did on these synthetic sequences. So then the question is, does this actually mean that synthetic sequences are bad to use? We argue not, because one thing, and we'll get to a second one later, is that you can use this for transfer learning. You can actually start training on your much larger set. In this case, it was 280,000 or so of synthetic sequences, and then add actually the natural sequences to that. And this combined model actually gets a better, like 0.86 now correlation instead of 0.8 on the actual natural human sequences. But what we're now doing, actually, now that Fred stayed as a grad student in the lab, and I see Carlos, I think, somewhere in the back here, so it was proposed by Carl de Boer and others in the community to really use what's called now gigantic reporter assays with lots and lots of completely random sequences, because it allows you to explore your sequence space outside of the small fraction that evolution makes us available, essentially. And it also has a simpler makeup to learn the rules. Right, natural sequences are often overloaded with all kinds of stuff. I mean, you would have RNA binding proteins and G quadruplices and all kinds of stuff in a 5' UTR. But in random sequences, the chances that they all overlay and make your inference hard is much smaller. And of course, they're super cheap. So you can actually generate millions and millions of reporters, essentially the numbers of cells that you can assay, and the sequencing cost is the limitation and not the cost of reporters. And of course, the con is that the sequences may be too far from functionally relevant space, but you can actually then use only parts of the library. And I just want to illustrate this here. This is sort of the GC distribution in natural UTRs. They're GC richer because they're next to promoters that often overlap with CPG islands, and that's what you can see in the UTR as well. And so we can actually, just by changing the GC content, match that distribution better than using fully random UTRs. And so this is what the strategy that we're now using, and actually going in, having redesigned the reporters to actually have the first libraries now with about 10 million reporter constructs. So scaling up essentially immediately by 2 orders of magnitude from the original publications here. And then hopefully in this iterative way, learning how we can build better and better models, and also use these models then for sequence design. Okay, I'll skip a couple of things and just want to briefly mention the second project, so Katja's work. So this is synthetic work, and that hopefully helps us to understand how sequences are made up and what are the important sequence features, also to construct UTRs or regulatory sequences with design properties. But how do we annotate active open reading frames? So we already saw ribosome profiling is one way to do that, but there are variants of this protocol that are actually better at that. You can use different drugs, for instance, to inhibit the ribosomes at translation initiation, like harringtonin or damn, lactimidamycin. I knew it. Or you can also profile ribosomal subunits. So there's a really funky protocol that I just want to show you called TCP-seq. It's this here, Translation Complex Profiling, by Thomas Price's lab, originally published in yeast, but there's also data on human and some other follow-up publications. And the idea there is that you actually fractionate out different complexes of the ribosome, and that you actually see the fragments of the scanning ribosome along the 5' UTR. So this is the small subunit that's actually scanning along here. And then if you align your reads, you actually get a two-dimensional profile by read length versus position of where it aligns, because essentially the small subunit will stop as soon as you hit the start codon, and it starts like building the complex of translating ribosomes, the fully elongating ribosome. And so you see these diagonal read profiles aligning. And so this is a 2D picture of read alignments. Same thing here in the human data, it was published later. And for that, we can build simple image convolutional networks on these patterns to now just without any sequence context or with the difficulty of the regular ribosome profiling context, really directly read where did the small subunit stop and where did it start assemble a fully functional ribosome. And so how we do this is that we essentially have a positive window in a certain size range and distance range aligned essentially in the middle of our candidates of our reference position. And then we create negative sequences directly around because we want to mimic essentially the ribosome scanning. I don't want to select some random sequences. I really want to mimic that process that the ribosome scans and stops at a specific point. And so that's our negative data. This is actually right next to the start codons. And with this, we build a neural network. Here you can just see how it trains essentially. In the yeast data, it actually really nicely stabilizes. It doesn't overfit. And if we look at the patterns against with this attribution methods with the explanation, you can see that in different quartiles of high, medium, low, or tertiles, I guess, of expression, you see that you can capture these patterns essentially of these read alignments in 2D. Yeah? And so that means if we actually take all the data, especially the ones with the lower, from the lowly translated genes, we get the most robust predictions. That's what you see here. If you train not on the highly covered genes, but on the ones with lower coverage in yeast, you actually get the most stable prediction across different expression classes. In human, unfortunately, that's not the case. Human, of course, is, I don't know, definitely more complicated, maybe also smarter than yeast. Here, we have to stick with the high to medium covered genes because you can see that this profile and these pictures really start to fall apart. And we do, however, are able now, and this is what we can see here, to annotate also these upstream open reading frames and also open reading frames with alternative start codons, so with non-AUG start codons, for which you see here, actually the profiles, they also look nicely because translation starts don't always start at AUG. There's also different start codons, CUG, GUG, and so forth that can be used. And with these methods, we're getting closer, combining them with the sequence context around especially these difficult start codons to learn combined models that can hopefully realistically predict where all these active, in fact, potentially regulatory elements actually are. And to wrap up an ongoing work here, we're actually now switching, because it's easier, to these drug treatments, to Harringtonian treatments specifically with our lab next door. And this lab has created transgenic lines with different AIDs, auxin-induced degrons, for these different initiation factors. So then we don't only see how does the translation get perturbed when you mess with the translation factors, but also how they might shift around, because some of these alternative initiation factors are, for instance, responsible for cap-dependent versus cap-independent translation. So with that, I want to wrap up, point out again, Fred and Katja, who did most of the work that I talked about today. Of course, like everyone, we're looking for computational postdocs, and I'm happy to take questions.
Okay, just one burning question.
Right, very nice talk. So I'm wondering, beside looking at the primary sequence motif of those orbs, have you checked like secondary structure, things like that?
So the RNAs, the
the-
RNA secondary structure.
Yeah.
That's something that we want to do, especially with these longer reporter sequences. So in 50 nucleotide reporter sequences, I think Quaid's the absolute expert. There's of course very little folding. And so that's exactly why in our larger reporter libraries are going to 200 nucleotides, and then can hopefully also see the effect of the secondary structure, because once you translate the UORFs, you might actually disrupt and change the secondary structure and, for instance, make the main start codon accessible. Yeah. Thank you.